Okay, guys, in this lecture, we will be discussing about the cell biology. This is an animal cell and this is a plant cell. If we look at the differences between the animal cell and the plant cell, animal cell does not have a cell wall, whereas plant cell, it has a cell wall, also has a cell membrane, but animal cell, it has only cell membrane. The nucleus in animal cell is almost centrally placed, whereas nucleus in the plant cell is not centrally placed. Well, in both the cells, like the animal and plant cells, mitochondria are dispersed throughout the cell in the cytoplasm. Centrioles are present in animal cells. And chloroplast is a distinct feature of the plant cell. Ribosomes are present in both the cells. Then the endoplasmic reticulum is also present in both the cells. The nucleus, it is covered by the nuclear membrane. Nucleoli are also present in both the cells. Chromosomes are present in the nuclei of both the cells, whereas in plant cells, the centrally placed large vacuole is present. On the other hand, in animal cells, the small vacuoles are dispersed over the whole cell. They are not centrally placed, and there are many vacuoles. Golgi apparatus is present in both the cells. Cytoplasm is present in both the cells, etc., etc. Prokaryotes, unicellular animals, or even like we can say that bacteria, they have a nucleus, which is basically a single circular chromosome, not actually a nucleus, but the centrally placed genetic material. Cyanobacteria, bacteria, and rickettsiae, these are the examples of prokaryotes. Eukaryotes, complex cellular organization, there are membrane-bound organelles, well-defined nucleus. They are present, these type of cells, they are present in higher animals, plants, fungi, and protozoa. If we look at the differences between the two cells, like the prokaryotes and the eukaryotes, both are animal cells. This is basically very primitive cell, bacteria. And they have even the cell wall. Cell wall is not present in eukaryotic cell. They have cell membrane and the centrally placed nucleoid. Cytoplasm is also present, whereas there is a very complex structure of the eukaryote cells. Cellular function for movement, conductivity, metabolic absorption, secretion, excretion, respiration, and communication. Here we can see one complex animal cell, different types of endoplasmic reticulum, the smooth and the rough, Golgi apparatus, centrally placed nucleus, nucleoli, etc., etc. Ribosomes. Eukaryotic cell. The nucleus of eukaryotic cell, it is the control center of the cell. It has a double membrane, contains chromosomes and nucleoli in it. We are talking about the eukaryotic cell, and there is nucleus, nuclear envelope, which are membrane around the nucleus, nucleolus. It is having organelles involved in the cell division, DNA, chemical blueprints of life, DNA replication, semi-conservative method, repair with nuclear enzymes in some cancers, sometimes defective repair enzymes, and transcription making mRNA and tRNA and rRNA from the DNA gene templates. Histone proteins bind to outside of DNA, double helix, and protect DNA, as well as control what genes are expressed at what time. Cell division through mitosis. There are different phases of mitosis: prophase, metaphase, anaphase, telophase, and interphase, the longest phase. We call it cell cycle, which we are going to discuss in a separate lecture. Eukaryotic cell, we are discussing functions of nucleus. Nucleus is the house for most of the cell's genetic material, the DNA and RNA. The nucleus is surrounded by a porous membrane known as the nuclear membrane. The RNA moves in and out of the nucleus through these pores. Proteins needed by the nucleus enter through the nuclear pores. The RNA helps in protein synthesis through transcription process. The nucleus controls the activity of the cell and is known as the control center. The nucleolus is the dark spot in the nucleus, and it is the location for ribosome formation. Cytoplasm. There is cytoplasmic matrix. The matrix maintains the pressure of the cell, so it ensures the cell is not going to get shrunk or is not going to even burst. Viscous fluid, like cytoplasm, is a viscous fluid containing organelles. Components of the cytoplasm, different components in different cells, interconnected filaments and fibers. Fluid is cytosol. Then there are organelles, not the nucleus, and storage substances. Here we can see the cytoplasm. Ribosomes, organelles. rRNA made from DNA and two protein subunits. Free ribosomes, they are independent from the reticulum, and attached ribosomes, they are associated with the endoplasmic reticulum. Endoplasmic reticulum, ER membrane complex inside the cell. It is the site of protein synthesis. There are two types of endoplasmic reticulum, which I have already mentioned: the smooth and the rough endoplasmic reticulum. Endoplasmic reticulum bound to the ribosomes, it appears rough, so we call it rough endoplasmic reticulum. Whereas the smooth endoplasmic reticulum, it does not have the ribosomes. This is the difference. Eukaryotic organelles, ribosomes involved with protein synthesis, line up tRNA and amino acids, rRNA and mRNA with enzymes that attach amino acids into long polypeptide strands. Endoplasmic reticulum, site where ribosomes, they produce the proteins. We have already discussed about it. Eukaryotic organelles, Golgi complex, final processing site for proteins, especially those destined to be secreted from the cell, like hormones or the enzymes. Golgi bodies are the packaging center of the cell. The Golgi bodies modify the molecules from the rough endoplasmic reticulum by dividing them into smaller units with membrane known as vesicles. They are flattened stacks of membrane-bound sacs. Proteins from the endoplasmic reticulum are packaged into the Golgi complex. Cisterna, and its plural are cisternae. So a cisterna refers to a flattened membrane disc that makes up the Golgi apparatus. A Golgi stack may contain anywhere from 3 to 20 cisternae, but most contain about 6. Golgi cisternae can be separated into 4 classes: cis, medial, trans, and TGN, trans-Golgi network. Each cisternae class contains different enzymes working within. This helps to prevent any two reactions from competing. Almost all cisternae in the Golgi apparatus present multiple stacking. There are only 8 instances in which the stacked characteristic has been lost. Function of cisternae. Cisternae pack and modify proteins and polysaccharides. Biosynthetic cargo proteins travel through cisternae and undergo glycan remodeling, and other modifications, cisternae package the proteins and then send them to transport carriers. They also package polysaccharides that are synthesized in the Golgi apparatus. The compounds enter at the cis phase of the Golgi stack and exit out the trans side where most of the packaging occurs. Cisternae. This is the Golgi complex. Golgi complex usually associate with endoplasmic reticulum and outer cell membrane site of final processing of proteins to be secreted out of cell. Lysosome membrane bounded structures, they originate from the Golgi and contain lytic enzymes. Digestive system of the cell it is composed of. Catalyze and break down proteins, lipids, nucleic acids, and carbohydrates. It has a role in the autodigestion and detoxification of the cell. Some vitamins can destabilize lysosome membranes. Retin A anti-wrinkle cream and cortisone stabilizes membrane, preventing the cellular breakdown, which is a part of anti-inflammatory action. If the lysosomes were not membrane bound, the cell could not have used the destructive enzymes. Peroxisomes. They contain oxidative enzymes, break substances down into harmless products. They help in digesting long chains of fatty acids and amino acids and help in synthesis of cholesterol. Mitochondria. They are surrounded by a double lipid bilayer membrane, participates in oxidative phosphorylation, increased inner membrane surface area provided by cristae, location where enzymes of the Krebs cycle are located and where ATP is produced in the electron transport chain reactions. Mitochondria is the main energy source of the cell. They are called the powerhouse of the cell because energy, ATP, is created here. Mitochondria consists of inner and outer membrane. It is spherical or rod-shaped organelle. It is an organelle which is independent as it has its own hereditary material. This is the mitochondria. Mitochondria is metric. And here we can see the inner membrane. This is the outer membrane of the mitochondria. Vaults. The vault or vault cytoplasmic ribonucleoprotein is a eukaryotic organelle shaped like octagonal barrels whose function is not fully understood. Vaults are cytoplasmic organelles which under an electron microscope resemble the arches of a cathedral vault with 39-fold symmetry. Vaults become part of the lipid rafts where they may play a role fighting pathogens. Cellular trucks, they have a role in transportation of molecules such as mRNA from the nucleus to parts of the cytoplasm. It is also thought that vaults play a role in protein synthesis. So they play some role in the synthesis, but mostly in the transport. That's why they are known as cellular trucks. Cytoskeleton. These are the bones and muscles of the cell. They are considered as maintains the cell's shape and internal organization, permits movement of substances within the cell and movement of external projections. These are microtubules, the centrioles, and the microfilaments. This is how they are. Plasma membrane. It controls the composition of a space or compartment they enclose. Its structure, if we look into it, cavule plasma membrane pits that are abundant in many mammalian cell types play a role in remodeling the extracellular environment in response to plasma membrane stresses. Then lipids, amphipathic lipids, hydrophilic and hydrophobic. Phospholipids, which have polar hydrophilic heads and they have non-polar hydrophobic tails. So their heads are water-loving and their tails are water-repellent. Glycolipids and cholesterol and carbohydrates. Here we can see head, which is polar hydrophilic, and non-polar tails, which are hydrophobic. Proteins. These are the integral peripheral transmembrane like different types of proteins, their functions, they work as receptors they are being used for the transport purposes. They could be enzymes, they could be used as surface markers, and they help in adhesion as well. So here we can see this is the outside, and this is the plasma membrane of the cell. They can act as a transport channel. Then they also secrete some enzymes. They also work as the cell surface receptors for certain hormones, for some neurotransmitters, as well as for some medicines or drugs. Then comes the self-surface markers, and they help in cell adhesion, cell-to-cell adhesion. They are also helping in maintaining the integrity of the cell by attaching the cytoskeleton. Proteolytic cascades. Caspase-mediated apoptosis, blood coagulation cascade, matrix metalloproteinases cascade, and complement cascade. Apoptosis. Apoptosis is a process of programmed cell death that occurs in multicellular organisms. Biochemical events lead to characteristic cell changes, morphology, and death. These changes include blabbing, cell shrinkage, nuclear fragmentation, chromatin condensation, chromosomal DNA fragmentation, and global mRNA decay. There are two pathways, intrinsic and extrinsic. Both pathways include cell death by activating caspases, which are proteases or enzymes that degrade proteins. The two pathways both activate initiator caspases, which then activate the executioner caspases, which then kill the cell by degrading the proteins indiscriminately. Coagulation, also known as clotting, we have discussed it in one of the lectures previously on blood coagulation. It is the process by which blood changes from a liquid to gel, forming a clot. It potentially results in hemostasis, the cessation of blood loss from a damaged vessel, followed by repair. The mechanism of coagulation involves activation, adhesion, and aggregation of platelets, along with deposition and maturation of fibrin. Disorders of coagulation are disease states which can result in bleeding, hemorrhage, or bruising, or obstructive clotting, thrombosis. Matrix metalloproteinase cascade. Matrix metalloproteinase, MMPs, are calcium-dependent, zinc-containing endopeptidases. Other family members are adamelicins, then cerelicins, and estacins. The MMPs belong to a larger family of proteases known as the medzinsin superfamily. Matrix metalloproteinases are also involved in the destruction of periodontal ligament in case of periodontitis. Collectively, these enzymes are capable of degrading all kinds of extracellular matrix proteins, but also can process a number of bioactive molecules. They are known to be involved in the cleavage of cell surface receptors, the release of apoptotic ligands such as the Fas ligand, and chemokine, cytokine inactivation. MMPs are also thought to play a major role in cell behavior such as cell proliferation, migration, adhesion, and dispersion, and in differentiation, angiogenesis, apoptosis, and the host defense. Complement cascade. Complement cascade is a part of our immune system that enhances or complements, that's why it's called complements it, the ability of the antibodies and phagocytic cells to clear the pathogens from an organism. That's why it is known as complement system. It is part of the innate immune system, which is not adaptable and does not change over the course of an individual's lifetime. However, it can be recruited and brought into action by the adaptive immune system. The complement system consists of a number of small proteins found in the blood, in general synthesized by the liver and normally circulating as inactive precursors. Proproteins, when stimulated by one of several triggers, proteases in the system cleave specific proteins to release cytokines and initiate an amplifying cascade of further cleavages. The end result of this complement activation or complement fixation cascade is massive amplification of the response and activation of the cell-killing membrane attack complex. There are three pathways. One is the classical complement pathway, the other one is the alternate complement pathway, and third one is the lectin pathway. Membrane fluidity. Cell-to-cell adhesions. Adhesions of like cells is a primary feature of the architecture of many tissues. Cell adhesion occurs from the action of the transmembrane glycoproteins, which we call cell adhesion molecules. Selectins, integrins, syndicins, and cadherins, these are the examples. Cellular adhesion is essential in maintaining multicellular structure. Cellular adhesion can link cells in different ways and can be involved in signal transduction. Cell adhesion is also essential for the pathogenesis of infectious organisms as well. Different types of cell-to-cell adhesion. Integrins, cadherins, etc., etc. We are discussing cell-to-cell adhesions, cell junctions. These are different, like desmosomes, tight junctions, and gap junctions, and then gating. Here we can see the junctional complex. Cellular communication. It could be through the direct link up, through the gap junctions, through hormones or neurohormones. Then it could be paracrine or autocrine, and through the neurotransmitters. So this is the gap junction. There is a gap between them and they make the gap junction. So the small molecules and ions, they can be exchanged. This is the transient direct link up of the cells. Here we can see the paracrine secretion. This is the cell which is producing some secretion, and it is secreting it, which is reaching the target cell. This is the neurotransmitter secretion. The neuron is producing some kind of neurotransmitter, which is then going to the target cell, and it is affecting the target cell. This is hormonal secretion. Hormones are produced somewhere else in the endocrine system. Then through the bloodstream, they are reaching the target cell, and they are affecting that. Over here, again, we have the neurohormonal system. The neuron is producing a hormone which is being transported to the target cell by the bloodstream and is affecting over there. Signal transduction. Extracellular messengers, they convey instructions to the cell's interior, transfer, amplify, distribute, and modulate. Channel regulation, second messengers, two pathways are there: adenosine monophosphate or CAMP and calcium ions. Here we can see signal ligand receptor. This is the plasma membrane. So there is a modulation by other events. There will be relay, then there will be an amplification. After amplification, there will be divergence, regulation of the gene expression, changes or regulation in metabolic pathway, or changes in the cytoskeleton may occur. Metabolism, cellular metabolism. Chemical tasks of maintaining the essential cellular functions. Anabolism, that is energy using process, and catabolism, that is the energy releasing process. Adenosine triphosphate or ATP. It is created from the chemical energy contained within organic molecules used in synthesis of organic molecules, muscle contraction, and active transport. Universal fuel for all cell functions is ATP. The electron transport chain, cytochromes are involved with the production of ATP. Digestion. Glycolysis, first step in the breakdown of glucose, which is a 6-carbon sugar, and then it is going to be converted into pyruvate, which is a 3-carbon. So 2 molecules of pyruvate are being produced from the 1 molecule of glucose or 6-carbon sugar. Well, there is anaerobic, no need for oxygen, like mitochondria reactions. If no oxygen is present, pyruvate will be converted into lactate. Citric acid cycle, also known as Krebs cycle, we have discussed it in a different lecture. The Krebs cycle, or the tricarboxylic acid cycle in the mitochondria. Then oxidative phosphorylation, it occurs in the mitochondria. Here we can see when we ingest the food, either proteins or polysaccharide or fats, then proteins are being digested in the phase one, they become amino acids, and polysaccharides, they are being converted into simple sugars, fats into the fatty acid, and then phase two, intracellular breakdown of the subunits to acetyl-CoA, accompanied by production of limited ATP and NADH. Then finally, there will be phase three, production of NADH and yielding ATP via electron transport, waste production of water and carbon dioxide and ammonia, and urea is going to be excreted. We have discussed it in our citric acid cycle as well as in our urea cycle as well. There are some detailed lectures which we will be discussing the membrane transport, electrical impulses, the action potential, and cell cycle. Tissue formation, founder cells, chemotaxis, contact guidance, and cellular reproduction. Tissue formation, the founder cell, then mitosis occurs, recognition and adherence of specialized cell junctions. They are attached to the basement membrane. Just giving you an example of the epithelium formation. Types of tissue. Epithelial tissues, they could be simple. Simple means having one layer, stratified meaning having more than one layers. Squamous, cuboidal, columnar, pseudostratified, then structures with the cilia and microvilli. We have discussed it in detail about one of the lectures on epithelium. Connective tissues, ground substance, fibers, loose and dense connective tissue, elastic and reticular connective tissue, cartilage, bone, vascular and adipose tissues, they all fall under the category of connective tissue. Muscle tissue, smooth muscle, striated or skeletal muscles, and the cardiac muscles. So over here, we are done with our lecture on cell biology.